Welcome back. Did everyone find a nice scenic place to enjoy lunch? It's hard to find a bad spot to enjoy lunch here. So we are now into the afternoon of our program, and this is the second of 3 capacity building sessions for today. If you were with us for our last session, you'll note that the format of this session is a little different. There are tables around the edge of the room, which will be part of our session. So if you're just hanging out there, you might want to come join us up here instead of sitting back at a table. We have 3 speakers that will give some presentations about seed banking and conservation horticulture. And then I referenced this sheet before. Was in your folders. It gives a list of the speakers and also has a list of what's called info tables. So you'll see that there are 4 information tables: supplies, materials, field collection for conservation horticulture, hand pollination, micropropagation, tissue culture, etc., collecting and submitting seeds for banking, seed storage techniques, and data collection forms and databases. So after the 3 presentations, there will be hosts sitting at each of these 4 tables, which are in the perimeter of the room, and you'll have time to go to 2 of those tables that most interest you to get some deeper dive information on those topics. So we'll start with presentations, and then I'll explain again, rotating to these tables later after our presenters speak. We will have opportunity for Q&A as well, and I think that that's enough from me, so I will introduce our first speaker, Lisa Ellis, Deputy Chief of the Division of Restoration and Recovery for Fish and Wildlife Service, Southeast Region.
So policy isn't necessarily all that exciting, so I apologize for a lot of regulatory talk. But the controlled propagation policy for federally listed species was passed in 2000, and it's designed to address the role that controlled propagation plays in conservation and recovery of listed species. It provides guidance and establishes consistency for the use of controlled propagation, and it assists with smooth transitions between conservation efforts, such as captive breeding to reintroductions. So the policy addresses controlled propagation for the purposes of preventing extinction, recovery-oriented scientific research, maintaining genetic vigor and demographic diversity, maintaining refugia populations, providing individuals for establishing new self-sustaining populations, as well as supplementing and enhancing extant populations. There's a number of goals of the policy. They include coordinating recovery actions specific to controlled propagation activities, maximizing the benefits to listed species, assuring that appropriate conservation measures and other controlled propagation and other existing recovery priorities are considered in making decisions, and ensure prudent uses of funds. So the scope of the policy is for establishing and maintaining refugia populations, producing individuals for research, technology, development needs, producing individuals for supplementing extant populations, and individuals to reintroduce in a suitable habitat within the species' historical range. So there's a whole lot of overlap with the goals and the scope of the policy. It's very, very focused in on the scope there. So this policy actually applies to candidate species, proposed species, as well as listed species that are indigenous to the United States and its territories. The species that the service has or intends to prepare recovery plans are what's supposed to be addressed here as originally written in the policy. That has been relaxed a little bit. We often use recommendations from 5-year reviews or other documents to establish these types of programs. So the policy has 14 points, and I'm going to run through those points just because that is essentially our policy. And these are paraphrased. If you look at the actual policy, there's a lot more information there and a lot more detail. But for control population, it'll be used as a recovery strategy only when the measures to maintain or improve a species in the wild have been or would be insufficient to achieve recovery. So essentially, we use this policy when recovery would be difficult without taking the individuals into captivity and doing controlled propagation. Not necessarily as a last resort, but definitely not the place you want to start when you're putting together a recovery program. Controlled propagation should be coordinated with other conservation actions and recovery measures, so this shouldn't be done in isolation. This should be done as part of a coordinated overall program for recovery. And it should be based on recommendations in an approved recovery plan whenever possible. As I just mentioned, we've relaxed that a little bit, and it doesn't necessarily need to be in a recovery plan. It can be something that's recommended in a 5-year review or another recommendation, or even simply an approved process. So the fourth point of the policy is that controlled propagation is based on specific consideration of potential ecology and genetic effects of removal from the wild. So removing those individuals from the wild, what does that do to the wild population? And this can be done as part of a Section 7 consultation or a Section 10 permit, that analysis to make sure that what we do when we take individuals into captivity isn't affecting the wild population. It should be done to conserve genetic variation and species integrity. Under the policy, no intercrossing is allowed unless covered in a recovery plan and recommended. Again, like recovery plans in general, if there's another service-approved policy out there for this individual species recommendation, then it is allowed. Controlled propagation should also have a genetic management plan developed if feasible. Obviously, there's a lot of variation in how much information is out there on different species. So for some species, this is feasible. Other species, it isn't. But we want to make sure that the genetic makeup of propagated individuals is representative of the wild population, and that the propagated individuals are suitable for reintroduction. We don't want to have artificial selection on those individuals that we reintroduce. The seventh point of the policy is that controlled propagation should be conducted in a manner that takes all known precautions to avoid the introduction or spread of disease and parasites into the controlled environment or back into the wild when they're released. It should also be conducted in a manner to prevent escape or accidental introduction outside the historical range. Often when we're doing these programs where we're doing controlled propagation, we actually move them out of their historical range. So if they're being bred outside of the historical range, you want to make sure that you're taking the precautions that they don't get reintroduced to that new area, or introduced to that area. It's also recommended that the controlled propagation should take place at multiple locations in order to reduce the risk of catastrophic loss. And this is especially important if you're taking a substantial fraction of the species or a unique population into captivity. You don't want all your eggs in one basket, basically. So 10th, I think everyone is fairly good about doing. It needs to be coordinated with appropriate organizations and individuals. And this includes not only other federal agencies, but state agencies, nonprofit, academics. Obviously, we have a lot of partnerships with zoos and botanical gardens, so those are often front and center in terms of coordination. The 11th point is that we need to meet our information needs in accordance with accepted protocols and standards. And that's referring to record keeping, making sure that you have a stud book for some animals, or just records of various life stages, tracking which seeds came from which location, that type of thing. Okay, almost done with the 14 points. The 12th point is that controlled propagation will be implemented only after a commitment of funding is secured. That's not always a realistic goal. A lot of the times we do start before we have that funding secured. The 13th point is that an established recovery or reintroduction plan needs to be in place unless it's covered in a recovery plan. Sometimes in recovery plans we go into a lot of detail on reintroductions that are necessary. And that helps us allow for evaluation of progress on the project as well. Controlled propagation and reintroduction plans should identify measurable objectives and milestones for the effort. They should include protocols for health management, disease screening, monitoring and evaluating characteristics such as genetics and phenotype, and of course record keeping. And it should be conducted in accordance with all applicable laws such as the Endangered Species Act and NEPA. So for plants specifically, the policy states that the service and others involved in controlled propagation are encouraged to follow the protocols and standards of the Center for Plant Conservation. And for animals, I think it's AZA standards, so we don't make it a requirement, it's just encouraged, but from my experience, pretty much everybody does go by these guidelines. And they have a variety of guidelines, including genetic sampling, how to develop a reintroduction program, seed storage, collection management. And for example, like the genetics guidelines includes information on how many populations you need to be sampling in order to get the representation, and how many samples per population should be collected. So they're great resources for setting up a program. The policy also goes over risk evaluation. And this isn't a requirement, but it's recommended, and whether it's formal or informal, there are a number of things that the policy wants you to be thinking about. And that's the effect that the removal of the natural parental stock may have on the increased risk of extinction in the wild. Again, this goes back to ensuring that we're not causing extinction in the wild. The potential for equipment failures and human error, disease, or other catastrophic effects where you're housing the individuals that you've taken into captivity. Any potential increase in inbreeding or other adverse genetic effects. The potential effect of exposure to a novel selection regime, especially if you're moving them indoors or outside of their historical range. And then the potential loss of genetic differences between populations as a result of mixed stocks. And the list goes on. There's lots of things that you should be thinking about when you're making these decisions. So some of the caveats and cautions that go along with this policy are, the first priority is always to recover the population and the natural habitat in the wild. Recognizing that oftentimes we get into a situation where that's not possible, or we're in a spot where we maybe can, maybe can't, and that's the point where you start talking about controlled propagation. So, and this also does not substitute for addressing the factors responsible for the status of the species. In other words, the threats. You still need to be out there working on the ground, reducing threats, making sure that habitat is preserved, both for reintroductions and to be conserving the populations that exist in the wild. So one of the things that I mentioned of the 14 points was that you need to provide us with some information collection. And I didn't go into a whole lot of detail on that particular one in terms of what needed to be provided to us, and that's because we stopped collecting extensive data in 2004. So I do have some data from 2004 and just a little bit from 2013, just in terms of the prevalence of the use of the policy, and it's pretty interesting, so I thought I'd share it even though it's out of date. Nationally, we have, out of about 1,500 federally listed species, there's about 650 of them that are under some sort of controlled propagation program, and that actually is 2013 data. The rest is 2004. And about 90% of those 650 species under controlled propagation are endangered species. So that does indicate that it is being used for higher risk species. And although all taxonomic groups are represented in the controlled propagation programs, a lot of them are plants. Plants have a higher representation, so it's definitely being applied more commonly with plants. So we've also got over 1,000 partnerships represented in these programs, and over half of the species actually have more than one partner. They have multiple partners, and these range from other federal agencies, botanical gardens, states, universities, nonprofits, private landowners, and many others, including, for some of the species, Canada and Mexico. So some of the other information that we have on the purposes from the 2004 data is what these controlled propagation programs are addressing. And for most species that are under a controlled propagation program, they're being conducted for multiple purposes, which is why these don't add up to 100. So we've got refugia, research, reintroduction, augmentation, education, and we do it for other activities that are in line with the purposes of the Act. So for examples, I wanted to present some examples, and you'll notice that the big high-profile programs are all for birds or mammals. There aren't a whole lot of plant ones. And they tend to be smaller scale projects. And what I did was I went out to some of the botanists in our region and got examples that they have projects they either have worked on here in the region or when they were in other regions, just to present an idea of the type of things that have been done with plants or are being done with plants. But almost all projects, regardless of whether they're big or small and regardless of what species they're working on, they rely heavily on partnerships. That was one thing that is loud and clear, both in the big ones and the smaller ones. So one example that I got from Vivian down in our Panama City office is Harper's Beauty. And she did a study with the University of Florida on a germination study of Harper's Beauty and found that seed viability is high, about 80 to 90%, and that it's more likely following exposure to winter and spring temperatures. And seed viability in ex situ is not a limiting factor, but we don't know in the wild what it is, so that's unestablished. And one thing that Vivian was able to provide was the cost. So $25,000 plus staff time. If you look at that, that's doing a lot of good for the species at not a very high payment. So these are the types of studies and projects that we can easily, or should hopefully easily be able to find the funds for and make it a priority. Another one that I got from Pete Padavina in our Athens office, and he's been working on this with a lot of partners, was a couple of quillwort species that are granite outcrop species impacted by aggregate mining. And they went and they created some artificial pools and collected plants or spores from the wild population, grew them in captivity, and then placed back into the restored habitat that's been very successful in still maintaining the species. The last one I got from Mara Alexander from our Asheville office, and this is when she was working in one of our Region 2 offices with Texas wild rice. And this one had a couple different purposes. It was maintaining a refugia population, and it was also doing a genetic study to determine the degree and distribution in the wild. And they managed the population in order to maintain the genetic variability found in the wild populations. So that's just a few examples of the types of programs we have being done by biologists. Any questions?
We do have a question. Sorry.
No, you're fine. I was just wondering if you maybe could elaborate on why they stopped taking all that data after 2004, if you had any idea.
Why we stopped collecting it?
Yeah.
I think it was just considered a burden, both to the people that were providing it and trying to manage it. And we got 4 years worth of data, and I guess they felt that they had a pretty good handle on what was going on. We do still collect data, just not to that extent. And there were a lot of interesting things there to me, so I wanted to share the bigger database.
Are there plans to start collecting data at a later stage?
Not to my knowledge.
Are there any other questions? Well, thank you, Lisa.
Thanks.
Our next speaker is Johnny Randall, the Director of Conservation with the North Carolina Botanical Garden.
Well, thanks for having me. I guess my microphone's on, isn't it? This is a great conference, and I think things are going great. So today I'm going to talk about ex situ conservation. A lot of what I'm going to say, Joyce Mashinsky introduced earlier today, and Lisa has introduced some of this information as well, so that's good. We're building on each other's presentations. But I also want to point out that this presentation is built on a body of knowledge that has been accumulated mostly through the people who work through the Center for Plant Conservation. And so the materials that you're seeing today are not uniquely mine, but represent this, this body of knowledge. And also I want to quickly point out that ex situ conservation is really designed to help support in situ conservation, because there is no substitute for in situ conservation. So ex situ is great for augmenting populations, for reintroducing populations in secure sites, etc. So I'll talk a little bit about that as well. And so this is the basic definition of ex situ: protecting species outside of their natural habitat to areas equipped for the protection and preservation. And as Joyce Mashinsky pointed out earlier, this body of knowledge is embodied within these four not textbooks, but these four proceedings from symposia that were hosted by the Center for Plant Conservation. And pretty much everything you always wanted to know about plant conservation but were afraid to ask is in these books. So if you haven't looked at these, please do, because it's all there for the most part. Some of it's so this one is, this was the first one. There are certainly more genetics information out there than then, but still the basic premise and information here is completely valid. And there is this ex situ, in situ continuum, and I'm not going to go through all these bubbles, but down on this axis, there assumes that there are facilities to make these happen, and then ongoing efforts, whether it takes not too much of your time versus whether it is a high intensity type of situation. So for in situ, or for ex situ, where you put seeds in cryopreservation, you know, they're there for a long time. It's certainly not cheap to do that, and you need the correct facility, but it doesn't take much staff time to do that ultimately. Whereas if you are trying to do specialist cultivation in a controlled environment that's been talked about earlier, where you make sure that plants aren't interbreeding with one another and seeds are falling on the ground and you're getting hybrids or inbred individuals in your seed bank. So like I said, I'm not going to go through this, but there is this continuum between in situ and ex situ. And one element of this that isn't talked about much is this idea of inter situ, where you're growing a plant in a suitable habitat, not historical location, but that it can still respond to changes in the environment, and it is a conservation collection. But that has to be monitored carefully and can never be thought of, or at least it typically isn't thought of as a natural population, but rather as an experimental population. So what is ex situ delivering? So I'm not going to read all of these, but there's a lot of work going on out there among botanical gardens to collect and preserve the germplasm, either as seeds or as whole plants if the seeds aren't orthodox. And this is unfortunately true, especially in Hawaii, where there are more ex situ collections of wild populations of the rare plants in Hawaii. Hawaii is a true basket case when it comes to endangered species of all taxa. So, and a lot of efforts being put there. Every island on Hawaii has a different CPC institution and a seed bank, or at least a conservation repository. And we've heard a fair amount, or a little bit, about the Seeds of Success program through Department of the Interior, through BLM. And this is for collecting the more common plants of communities to help in restoration, to bring at least that matrix community back to some state where rare plants could ultimately be reintroduced to those sites. So the SOS program, I'll talk a little bit more in a bit. So why isn't ex situ delivering more? Well, some people think that it undermines in situ conservation. You know, if you've got all of these rare plants protected in a seed bank, why protect that last population or a population on a roadside when the Department of Transportation wants to widen it, for instance? Lack of professional confidence that people in botanical gardens can actually do this scientific research. However, I think that that is not so serious a problem anymore. I think that especially the Center for Plant Conservation Gardens have a very good track record, and they are very well respected. And also, you have to worry about, we've talked about liabilities before in terms of spreading pests and pathogens, and that's something always to consider. And I just turned my machine off. There we go. Whew. Okay. So determining ex situ needs in plant conservation. You know, is it appropriate? And if so, what techniques? We've already talked about cultivation a bit, seed banking as an insurance policy. Are there alternative techniques? Sometimes there have to be, like embryo extraction, and those can be cryopreserved. And should you think about translocation and reintroduction as part of this overall recovery program? Whoops. So there are clues whether or not you should undertake ex situ conservation or not. Small population size, or if you see populations declining precipitously, something needs to be done about that. Of course, we live in fragmented habitats, and a lot of times there's either the pollinator or something is missing, and you're not getting interbreeding and outcrossing species, so you have to take that in consideration, and that's where maybe augmentation on an in situ basis might be important. Variable environments, imminent threats, genetic or demographic problems, and if it's a high priority species that's not receiving as much attention as it might. So let's say it's a G1 taxon, but it's not listed under the Endangered Species Act. So that would be a situation where that would be the case. More clues. Plants aren't producing seeds. So this is a plant that we work with at the Center for Plant Conservation, Lysomachia aspergillifolia, and you know, there are whole patches of this growing in North Carolina coastal plain. They never produce seeds. It's rhizomatous. We're finding that this is probably one single individual in this, what looks like a population. So it is an obligate outbreeder. So when seeds are available, we do collect those. And we know that there are indeed different individuals in those populations. But you have to be very careful, and I'll go over some of the details of that seed collection. So these are just different clues on when ex situ is important. Who and what helps guide ex situ plant conservation? I'm not going to list all these groups, but there's a lot of them out there. So there are a lot of entities that are helping with these ex situ efforts, that provide guiding principles, support, financial assistance, etc. So there is, as Joyce pointed out earlier, there is this global strategy for plant conservation, and she talked about target 7. Was that it, Joyce? Or 6? At any rate, here's target 8. 60% of threatened plant species and accessible ex situ collections, preferably in the country of origin, and 10% of them including in recovery plans. And if you look at what's going on out there country-wise, this was a slide I got from Kay Havens. So if you look at the number of endemic plants in Brazil, for instance, there's a bunch of them. If you were to get 60% stored, 10% managed, that's a lot of seeds. And there's only 29 botanical gardens in Brazil to make this happen. Whereas if you go all the way down to the United States, 4,000 endemics. So with our 296 botanical gardens, we have the capacity to do this. And then there's another part of the continuum, whether a seed is orthodox or not. That's a big acorn, isn't it? What do you think that is? Mishohii. Exactly. And do you know what those little seeds are? Dutchman's pipe. Okay. So at any rate, there is this continuum between seeds that can be stored, can be dried and frozen, and ones that you can't store at all unless you could somehow extract an embryo and get it into cryopreservation. And as Joyce was mentioning earlier, do no harm. And that's one of the basic premises behind all of CPC's activities. And there is this problem with sample equaling trample. And it's something that I mentioned to all of the people I work with and all the interns that we have. And I've even had our interns take their shoes off to walk in barefoot in order to monitor a population or collect seeds. You have to have landowner permission. You know, it's very tempting not to do that, but it's really important because you lose credibility in your organization if you're trespassing on someone's land and you're taking their private property, and you lose credibility with your supporting agencies. Like, I have to spend tons of time getting permits to go on state parks and national wildlife refuges, and it's not easy. It takes a lot of time, but you have to do that. Otherwise, you lose all your credibility and you damage your partner's credibility as well, et cetera, et cetera. I won't go into all these. It's all fairly straightforward. So there's prioritization. Bill Brumback from the New England Wildflower Society is coming later today or tomorrow, and they are the organization that really got a lot of this jump-started, and they were one of the founding participating institutions at CPC as well. But Elizabeth Farnsworth has come up with, and this is great, you know, you should look at her prioritization scheme. She goes through a set of 3 decision matrices, and I'm not going to read all of these. I'll let you read those, but this is sort of a fine-grained approach whether or not you should go through ex situ conservation practices. Matrix 2, you consider G rank. It's rank in New England. And NEPCOP is the New England Plant Conservation Program. They have their own seed bank. And then Matrix 3, et cetera, et cetera. I'm not going to go through all of those, but these are all things that need to be considered before you undertake any sort of ex situ actions. So, sampling for diversity. There's a lot of detail here, and it's not simply going in and, you know, going out and collecting seeds and putting them in the glove compartment of your car, and then when you get home you put them in the refrigerator. That is not the way to collect seeds. So you want to capture genetic diversity, and you want to do it in such a way that you truly capture this diversity, and you want to treat the seeds very carefully. Why go to all this trouble if you're going to have a bunch of dead seeds by the time you get them into the freezer? How many populations should be sampled? Again, I'm not going to read all of these. There are protocols, and this is carefully laid out in the CPC literature. So, and I think all these presentations will be available too. That's why I don't see you busily scribbling down notes, I hope. So usually 5 or more if high diversity, limited gene flow, et cetera, et cetera. There's more things that, well, we'll talk about if destruction is imminent. How many individuals? Usually 10 to 50 or more, et cetera. However, for a seeds of success collection, you definitely want to get at least 50 individuals. But for CPC, you would ordinarily not take more than 10% of the seeds in any 10 years. However, for SOS, you would take at least 20,000 seeds from a population, but collected from usually at least 50 individuals. How many seeds individuals should be collected? Here we go, same things again. But if there is a planned reintroduction, you might step that up a bit because you're putting those seeds back into the mix. So multi-year collections, these are the things that you should follow: low annual reproductive output, high year-to-year variation in population size, and also to collect this genetic variability. It's the same thing if you're going out. This is sort of this example in a larger scale, but it's best to go out on multiple days. Plants aren't, it's not like wheat where it's all ripening at once. There's variation in ripening time. So if you only go out one time, you're collecting just one, you're immediately selecting for the the seeds that are from the earliest ripening plants. So Eric Minges, Ed Garant, and Hamas have suggested 3 rules. They've looked at this very carefully using population genetic models, population viability analyses, et cetera. So good information there from those individuals. And this is an exception if you're doing a rescue collection. So this is my son, one of my sons when he's a very young man. This is what that power line looked like when there were millions of Venus flytraps. But then look what happened when, I won't say who, went through with their plows and put a fire line in. So, and there's other federally endangered species in this particular power line as well. But we do rescue collections on occasion, especially if it's on private property. So here's some conclusions. You want to assess extent of population and estimate fecundity. Get these numbers. Collect as many seeds as you can, but not too many that would jeopardize the population. Sample in a stratified random fashion. High attrition rates are the rule rather than the exception. So you want to maximize your collection without over-collecting. Consider multi-year collection strategies, and then use an adaptive management type of, or adaptive collection tool as needed. So mostly we were dealing with orthodox seeds, and these are the basic principles for storing those seeds: to desiccate, to collect them, keep them dry in the collection process. Cool and dry is great, and until you get them to actual seed desiccation room or some apparatus that you have to desiccate them to the proper amount. And then store them at minus 18°C, your standard chest freezer temperature. What to do with recalcitrant seeds, the one that you cannot desiccate and freeze, like many of our tree species, like hickories and oaks, et cetera. Things like bloodroot and trilliums, you can't dry these seeds. So you have to take other measures, either living collections or through tissue culture. CPC has a great relationship with Valerie Pence at the Cincinnati Zoo and Botanic Garden. And these are some of the plant species that we have gotten into tissue culture through Valerie. And then there are, of course, the 4 horsemen of the in situ conservation collection apocalypse. And these are, this has to do, the first step, like I was talking about, that you take into that population to collect seeds, you are affecting genetic drift at that particular moment. You're only collecting at one particular time during its fruiting season. If you're growing plants out, this adaptation to cultivation, which is a real issue, mutation accumulation over time in small populations, and then simple attrition, like Joyce was pointing out. Some seeds last only 4 years, others much longer. When can unconscious selection occur? Okay, in the collection stage, the seed storage stage. Okay, some seeds have the ability to be desiccated, some not. Germination, you're not going to have 100% germination. So at every step of the way, you are reducing the genetic variability within that collection. These are some of the things you can do to minimize the genetic change. I probably need to speed up a little bit now. And there are seed banks, large and small, like the Doomsday Seed Bank at Svalbard, our own National Center for Genetic Resource Preservation in Fort Collins, Kew Gardens, and, small seed banks as well, like in that refrigerator. And I'll just give you a couple of examples of the seed collection we do at my botanical garden. So we have lots of seed collectors, especially through the Seeds of Success program. And it's a great way to double up on your resources. You know, if they're close to a rare plant population, they can collect there as well. If they're already 200 miles away from the garden, you might as well take advantage of that. And it's hard to tell. Well, you can't see the seeds, but anyone want to guess what Jake has collected with this piece of cloth? You would, huh? Seeds, sand spurs. So, you know, even sand spurs, they're really important for coastal dune stabilization. So that's one of our CPC collections. Not a rare sand spur, even though I'm sure there's a rare one out there. These are our facilities. They're meager right now, but we have plans. Alan didn't talk about this because he doesn't want to jinx it, but we're designing a herbarium building and plant research facility at our botanical garden, and it will have a state-of-the-art seed processing lab and storage facility, so we're really excited about that. But this is what we have now, which is an old office space that we sealed up, and it's humidity and temperature controlled, and that's where we desiccate our seeds, and we have a vacuum sealer in there that we can seal them up. So Seeds of Success, right now, we are in the third year, or actually going to the fourth year of a 3-year project through Seeds of Success. So these are our two partners, New England Wildflower Society and the New York Greenbelt Native Plant Center in New York City. So these are our 3 different zones. So this is the first time that SOS has ventured east outside of BLM lands. And we've been very successful in our work. And we hope that this funding can continue for Seeds of Success East. And these are just some of the totals this year for seeds that we've collected through our 3 different programs. I didn't mention also that we have, and have had for a long time, a seed collection program at our botanical garden, all native plants that can be made available to the public at their request. So no rare plants in that, no rare plants in SOS, but our CPC plants are. I won't go through this. These are some of our accessions over time, just going up to 2012. But this is interesting. You know, of course, you're going to forage closer to home. So nearly 50% of our accessions are from North Carolina, but our CPC range does go through other areas of the Southeast. So this is the different percentages that we have from other states. But we do cooperate with other gardens, and in fact, some gardens are using our seed bank at the moment since they do not have one of their own. So I'll leave you with these practical recommendations that are all fairly obvious, but still is something that we all need to think about. Thank you very much.
Are there any questions for Johnny? Oh, there are.
How important is the seed banking, and how can you link that, and how often we can be testing the viability of the seeds?
Okay, that's a good question. So you should always do an initial viability test to make sure that you are putting live seeds in the freezer. And you can either do germination or you can use tetrazoleum or something to tell if they're live seeds. But then you should be testing for viability every 5 years, in a 10-year period. So there are set times that you should be always testing those seeds for viability. And you should anticipate that if you vacuum sealed your seeds up, you want to have separate packets that are available for doing that viability testing that represent, and you can even bulk, because we collect, I'm sorry, I'm giving a long-winded answer, but you need to think ahead. So if you're going to do viability testing, you can ordinarily, you're collecting maternal lines in CPC collections. But if you're doing viability testing, you can have some bulk seed packets so you can get an idea of the germination across maternal lines.
Thanks, Johnny. real quick, I was thinking about in terms of the old paradigm was that we would keep different populations separate for fear of outbreeding depression. In managing seed lots and keeping track of those along maternal lines, how are you planning for in the future how those will be used or managed? So particularly in terms of outbreeding depression, we're finding increasingly that that was overvalued, and the fact is that we can actually benefit from merging those seed lots. Are you keeping track of those in a way that will be useful like that in the future?
Well, we still, of course, track everything by maternal lines, but so the general rule of thumb is that if you're doing an introduction to a site that still has individuals, you would augment that with individuals from that site. But if it's a novel site, you want to mix individuals from different populations. Did I answer your question? Okay.
So you are doing that? In that
Yes. Yeah. Yeah.
At this point. Okay.
But we haven't created any novel sites. We have only reintroduced things to historical locations or done augmentation. And Mike Coons will talk about some of that work later today.
Thank you so much. Our next speaker is Barbara Crane, the regional geneticist for the Forest Service Southern Region.
Thanks for having me here. We're going to switch gears just a little bit. We're going to talk about trees. And when Joanne and Jenny said, would you come and talk? I said, sure, but you're a botanist, and I'm a tree person. I'm not a botanist. I'm a forester. But I wanted to share with you what we're doing in the Forest Service with our partners as far as tree conservation, just to let you know what our activities are, because a lot of arboretums and conservation botanical gardens are getting more into tree conservation work, specimen items, etc. But also some of the protocols that we use may be helpful to you. Some of the guidance that we use could be used on your small native plants. So trees are native plants, but they're the 100-foot ones that we have to climb to get the cones and all the good stuff. So it's sometimes the botanist, it's a little bit easier just to bend down and rather than climb or shoot or something like that. So I'll talk about trees today. And I work in National Forest System. We're totally practical and applied. I do not work in research, and everything we do is on-the-ground restoration and reforestation. Okay, so trees will adapt, migrate, or die. What are we going to do about it? That's what I want to share with you today in my talking points. The drivers for tree conservation in our agency are projects, the tools and plans that we use and the ones where we have in development, and also, of course, partnerships. We always need partners because it's tree ranges go across many boundaries, many state lines, many regions. So why tree conservation for us? Well, we have to keep forests and forests as part of our mission in the Forest Service. But also, they're very long-lived woody species. We have trees that are 4,000 years old. Here in the South, we have some trees that are 300 and 400 years old. They're undomesticated, highly outbred, and very genetically diverse. Long generation times. This is a real challenge for tree conservation. I think, I'm not a botanist, but I'm sure with the small native plants, your generation times are probably maybe a couple years. And so my thought is, or the thought in the literature is, that the small native plants that you work with, they may be able to rebound to climate change and other drastic measures, whereas trees really can't. When they have a generation time of 50 years or 30 years or 15 years, they cannot respond that quick. So it's really important for us to pay attention to tree conservation and try to save some of these trees who will not be able to respond quickly like some other native plants can. Especially when it comes to adaptation. And then a lot of multiple factors that are affecting our forests. Everybody knows these typical culprits. WUI, climate change, forest health and disease, increased fire and drought here in the southern region. Probably in the southern region, we have 40 pests and diseases affecting our forests. So lots of challenges out there. We evaluate what tree species and populations are most threatened, and Johnny referred to some of these, of course, already. But looking at genetic specialists, populations that have low dispersal potential, low genetic variation, all these different fragmented populations, which is really bad for the southern national forests. If you ever look at the map, they're so disjunct and so fragmented. And so our tree populations have become the same way, a lot of our species. Populations at trailing end of the natural range, et cetera, et cetera. Climate change is a huge threat to our tree species, and trees cannot migrate as fast as the environment is changing. And I use that word fast loosely, but let me explain here. Trees can migrate generally 300 to 1,200 feet per year, and that's species dependent. But with the climate change and the rate the climate change is impacting our forests, trees need to be stepping it up 9,000 to 15,000 feet a year. They can't do it. So assisted migration, that's why tree conservation is really important to us. We have to be able to save these tree species that cannot keep up with climate change or can't respond and reproduce because some poor forest health pest disease is hammering them and killing them. We have to be there to help them do that. And so that's my job. I've been a forester in the South for 35 years, so I know a little bit about a lot of trees, and that's a lot of fun. Critical question then for us always is what's the most appropriate genetic material to plant now that will grow a healthy, sustainable forest in the next century? Our tree rotations are 100 years. Climate change has happened a lot quicker than that. And so our seed zones and planting zones are changing. USDA planting map has already changed. So we need to keep up with that for our trees. And when I talk about our trees, I'm talking about the genetic specialists. We have genetic generalists on some of our tree species. They're fine, they're robust, you never have to worry about them. It's a lot of the other tree species we need to worry about. So genecology research is really, really critical, and that is all about understanding what's happening to our trees in the environment, how they'll respond to climate change. Will they be resilient? Will they survive? Will they be okay? Will all their genetics kick in and they'll be fine? Or will their genetics not kick in and they'll kick the bucket? So this brings me to some of the tree conservation projects I want to talk about, in situ and ex situ. Again, just sharing with you what we do. The southern region, we have 140 tree species. It's like, holy crap, where do I start? You know, we have all across these different ranges and all these different geographic regions. And you can't do everything at once. We have a staff of 1 for the whole entire southern region, which is 14 national forests across 13 states and the Virgin Islands and Puerto Rico. So the staff of 1 is tired. That's why we have partners. So we came up with something called foregrass. Kevin Potter with FATAC, Eastern Forest Environmental Threats Assessment Center in North Carolina. He and I wrote this back in 2010. It was a way to rank all of our tree species. Look at the tree species that are imperiled now. Look at the tree species that we can wait for 5 years or 10 years or don't have to worry about. So, and this assessment it's real basic, really easy to understand. The botanists can use it. But we used all FIA data, all kinds of other information as far as population structure, et cetera, et cetera. It gave us the ability to rank the tree species. And from that ranking, we were able to identify 12 of our most highly imperiled species right now. And we focused on the southern Appalachians first because the mountains are more susceptible and vulnerable to climate change and other threats than the coastal and Piedmont species. And so there's the website for where foregrass is, or if you want a copy, just email me. But it's a real simple system. It's this Excel spreadsheet. You need to know a lot of data about your different plants, but still, it was really, really good system to use. So this is some of the output from foregrass. Here you see it. Here's some of the top 10 species we need to be working on now in the Southern Alps, and we are working on a lot of these. But in addition, because the southern region covers so many different forests and so many different ecological geographic regions, we're also working on all these other species right here, and you'll recognize a lot of these, and I'll go through some examples as I go through my talk, but lots to do out there. So red spruce in the southern Appalachians, really beautiful species. This is it's just fun to pull behind the truck and just get up there and get in the treetops that way. So we work with partners, Southern Appalachian Spruce Restoration Initiative. And if you look at the map here, the range map of red spruce, there's tons of red spruce up north. So why do we care about collecting these puny little things down here? Well, because in the southern Appalachians, those are at the edge of the glacial refugia. They may have genes still in their genome that has allowed them to survive and be more robust in the environment. So that's why we collect those. It's, you know, it might be the trick to, even though there's lots up here, we could still hang on to these because they're a lot more robust, genetically robust, if that makes sense. Table Mountain pine, one of my favorite species, up about 4,000 or 5,000 feet. We work with CAMCOR. They're the tree conservation co-op at NC State. They've been around for about 35 years. They do tree conservation all over the world. Well, they're helping us domestically in our own backyard with a lot of our tree species. And we are collecting Table Mountain pine. And the story with Table Mountain pine is it's a serotinous cone. It needs fire to open. And in the southern Appalachians, as the folks moved in and people moving south, they're not burning. So we're losing Table Mountain pine for the simple reason we're not doing prescribed burns. So we had to go back out and save that because there's only 30,000 acres left, and this is less, I mean, this is not even pure stands. And again, I know it's probably hard for you to think on the major scale like this, but this is how we have to think about when we talk about trees and talk about, 30,000 acres, that's not a whole lot. So here's a map of all the collections we've been doing, and we do range-wide collections all the way from North Georgia all the way up to West Virginia, New Jersey, and all the way up in there. So all up and down. And so when we decide to look at how we're going to collect some of our tree species, we try to collect at least 10 to 20 specimens per population. And then within each particular seed zone or state, we try to get 10 to 20 populations, and ultimately across the entire range, we shoot for 300 to 400 mother trees. And as we're out there collecting, we're collecting seed, but we're also collecting needle tissue. The needle tissue goes to NEFGEL, which is our National Forest Genetics Lab out in Placerville, California. They do the genetic analysis and look at the genetic diversity and make sure that we've collected enough of the genetic diversity of that entire species back on what we have in our seed bank. So we have just completed a 4-year collection project, and like Johnny mentioned, you don't collect just at one point in time, not just one snapshot, but year after year, because trees, sometimes they only have a cone crop once every 5 years, once every 10 years. It's not every year. I mean, loblolly, yeah, but that's a weed. When you're talking about these other really neat tree species, you have to go back. And then a lot of times, trees will have, in 1 year, if they have a real poor cone crop, the seed will not be very viable. So you have to keep going back to make sure you get viable seed in the first place. So there's a lot of differences with trees, and that's kind of what I want to share with you. There's a lot of challenges, but it's a lot of fun. Okay, make sure I got them all. Okay, Eastern and Carolina hemlock, I'm sure everybody knows that story. We've been collecting those two for about 10 years. Pitch pine, again, the same kind of story. If you look at the range here, the same kind of story. We have a little bit of glacial refugia left. There's more up north. But, if we want to reestablish pitch pine in our southern forest, according to our forest plans, which is the law, we need to make sure we have all these tree species collected in our seed banks so we can restore these species to the landscape. If pitch pine gets wiped out by a disease or a fire, we have the seed in the seed bank to do that. Atlantic white cedar, really neat species, really nice to be out and about on some of these coastal areas. There's not much of that left. It's been logged out. People want it for boats, for fences, for shingles on their houses. So we've done a lot of those collections, 3-year project with CAMCOR again. Butternut, lots of butternut up north of the Mason-Dixon, but we don't have a lot down here in the south, Tennessee, and a little bit in North Carolina, some in Kentucky and places there. But again, we want to restore it to the landscape. If the National Forest wants it on their land base, then we want to make sure we have the seed there to do that. So for those species, like Johnny mentioned, recalcitrant, that we can't do much with, the oaks, the hickories, American chestnuts, butternuts, et cetera, they can't be stored. You can maybe store acorns maybe a year. And that also another point about some of the conifers and other species, it varies by species and the biology is different for every tree. There's no one cookie cutter recipe. For example, longleaf pine seed will store about 5 years. Shortleaf seed will store about 10 years. Loblolly will store about 30 years. So it's different by species, and learning species biology as you're doing the collections and as you're storing is really, really important as well. So for our oaks, we have a lot of oak seed orchards we're establishing. We establish oak seed production areas out on the forest, on the districts. Scott Merkel is doing a lot at University of Georgia. He, like Jerry Pullman, is doing a lot of tissue culture work, cryopreservation of a lot of our sensitive tree species. Most of our tree species are referred to as sensitive, not threatened or endangered. I don't think very many of them are actually listed by Fish and Wildlife. Scott at UT is doing a lot of oak work for us. We have oak orchards, as I mentioned, across the range, because you got to capture all the different seed sources. You can't just collect one seed source and repopulate with that seed source. Because the southern region and the national forests are so broad and there's so many different states and seed sources, we have to make sure we capture as much as we can, because seed source is important to being locally adapted. So we wouldn't take a Florida longleaf and plant it in Texas. Don't do that. But I think you all know about seed sources. We have 7 species of oak in the southern region that are highly sensitive, and I think some of these might even be listed. We're attempting to work with some of them, and you know, for those of you who work with oaks, they may not have an oak acorn crop for 5 years, every 10 years. They don't start producing, some of them don't start producing acorns till they're 40 years old. So if they get wiped out by a disease before they even produce acorns, what do? So we're trying to be preemptive and proactive with all our species. Shortleaf and longleaf, these are our 2 workhorse species in the southern region. Longleaf, we've lost 97% of that ecosystem in those trees because of logging, and then replaced by conversion to other species that were faster growing. That loblolly weed again. 5 minutes? Thank you. And then shortleaf, we lost 50% of that ecosystem. Again, logging, and then planting with that weed loblolly. I don't like loblolly, can you tell? So we establish seed orchards and capture all the seed zones that we can in these seed orchards. And they're operational for reforestation and restoration. But one question I have, and we're not sure, is where do we establish our new seed orchards? Because of climate change, we have to make sure when we establish our new orchards, they're somewhere where the trees are going to be happy, they're going to reproduce. It's not where they are right now, because in 100 years from now, that site might be too dry, too hot, and they won't produce. So it gets to my third talking point about the different tools we have, the plans we have, et cetera, et cetera. Seed banking. We have a huge seed repository in Mississippi. Of all our conifer tree species, our recalcitrant tree species, we have standing seed orchards and standing seed production areas. The National Seed Lab, Vic is here. They test all of our seed for us, and we store it at less than 10% moisture content. Fort Collins, Johnny mentioned, National Center for Genetic Resource Preservation, we send a subsample of all our forest trees to them to store. So again, our seed is used in reforestation, restoration, assisted migration, which I talked about earlier. Some of these trees cannot keep up with climate change, and they're going to have to migrate. We're going to have to help them migrate to save some of our imperiled species. Seed biology studies are really important. There's a lot we don't know about our tree species. 140 species, you can't know everything. We know a handful about some of the tree species. Seed biology partners. Some of our seed will go to universities for research. I'll sell or donate some of the excess seed to the state people and share some of that seed. If Arboretum or Botanical Gardens want a specimen tree, I can send them some seed, donate some seed to them. Climate change, as I mentioned, again, it's a big issue for us. This is a practical guideline we developed in 2012. A bunch of the geneticists across multiple agencies wrote this. It's on the web. You can just Google it. And these principles are applicable to your little small native plants as well. And then other things we're working on, developing updated seed zones. There's a seed summit next summer, Forest Service and a bunch of other partners. We're talking about updating our seed zones, updating our planting zones. We're also talking about mixing our seed zones. So for instance, seed zone 7 and seed zone 8 on your planting map. Why not mix those seed zones so that one of the zones will survive now, then they'll start crossbreeding with each other, so that in 100 years, whatever's produced after that may survive as well. So you're mixing the genetics, hedging your bets, trying to make sure that they can survive. Capture is a national initiative. It's based on foregrass. We're trying to assess all our tree species across the entire country. We probably have 700. Right now we're working with 450. This is Kevin Potter again at FATAC and Southern Research Station, Gary Mann at the Washington office. Let me go to this slide. We had a big tree conservation meeting in May at Morton Arboretum. Lots and lots of partners, Pam and others were there. Everybody's aware of these huge big tree conservation initiatives that need to get done. We need to partner up. Kew Gardens was there. A lot of other folks. Everybody's on board with getting more tree conservation. So don't feel like you're not alone out there, because we're struggling through the same thing as tree people. But it was a really, really good conference, and good partnerships and good folks met there. Forest Service. So our plans, we have tons already in place. We have a plant genetics restoration business model, which looks at assessing, developing, and producing and conserving. And this is for trees and little native plants. Sorry, I got to differentiate. I'm always thinking trees. National Gene Conservation Charter, the National Genetic Strategic Plan, and at the moment I'm also working on a Southern Region Conservation Plan for our trees, but I'll also be working with our botanists if they need to go, if they want to entertain that, we can do that as well. And so this is my last slide. Partnerships are really, really important. We couldn't do it alone because, like I said, it crosses boundaries. We need to have everybody involved. National Genetics Lab in Placerville, great lab for doing all our genetic analysis. FATAC, which I mentioned, they look at future seed zones and ranges and climate change and all that kind of stuff. CAMCOR I mentioned also, the big tree conservation folks. Then there's tons and tons of other partners that we work with. And I'll end right there.
Any questions for Barb?